Yep. Second. Okay. So yes, thank you very much for that introduction, and it's really a pleasure to talk to you all today. Today I'm going to talk about a study that we been working on for several years and recently kind of completed mainly, where we've been using a sort of a large scale population genomic sequencing to understand the sort of history of species divergence and speciation, and also to try and understand the genetic underpinnings of morphological evolution in a pair of bumblebee species that are found in high altitudes in Colorado. So just to orient everybody about bumblebees, I just need to move this. Bumblebees are a social insect. They're important pollinators that are very important for pollinating mainly in more northern latitudes. They're a cold adapted species from the genus Bombus. There are about 250 Bombus species. They're haplodiploid, as are all other bees, all Hymenoptera, and they have a kind of interesting life cycle which differs from honeybees in that they build colonies during the spring, and which grow during the summer, and then eventually these colonies disperse, and mated queens hibernate over the winter on their own. So new colonies are built every summer. So, but for the point of view of this talk, the main important thing is that they're an important pollinator species, and so they've come under a lot of there's been a lot of public interest in bumblebees as the result of different studies that have shown that pollinators are threatened. In particular, in bumblebees, it seems that due to climate change, their distributions have been moving northwards as the planet is warming, and it seems to be that that they in the warmer regions, certain species reach the tolerance of the temperatures which they can survive in. Although it seems like the southern ranges are shrinking faster than the northern ones are expanding, which is obviously a worrying development, and also a reason why why it can be important to use genomics to understand how bees are adapted to different environments and how they might be responding to current changes. And another interesting finding, and also one of the finding that promoted the studies I'm going to talk about today, is that as well as the shifts in distribution, there have also been shifts in morphology. So tongue length is a very important trait in bumblebees, in that it's important for the mutualisms they have with plants. I mean, that's the tongues are what they use to extract the nectar, and it's generally considered that the bees with longer tongues specialize specifically on plants with longer corollas, whereas the shorter tongue bees are more generalist. And in the study that I'm presenting here, one kind of important finding was that over the last sort of 40 or 50 years, a reduction in the lengths of two different species has been recorded, which is inferred to sort of disrupt some mutualisms that occur between bees and the plants that they pollinate. So this is inferred to be something that could be quite damaging for biodiversity. But the kind of causes of these changes are not really clear, whether they're genetic or whether they're plastic changes. And so kind of the thing that started up the study I'm going to talk about now was trying to look at the genomics of these populations to try and understand whether there have been changes occurring in their genome. As you'll see, some of the goals changed as we went along, but that was kind of what got us started on these studies. So basically what we wanted to do was to generate some high quality genome assemblies of these two species from that are found in high altitudes in Colorado. That's Bombus sylvicola and Bombus spalteatus, which should facilitate genomic studies of a variety of different processes on these species, such as the genetic basis of adaptation and gene flow and population substructure, and other future studies. And so then what we wanted to do with this was to look at some of these, look at the structure of these populations, how they were connected, but also to try and understand what the genetic basis of the phenotypic shifts that were observed was, and if we could identify connections between genotypes and phenotypes using genome-wide association studies. So in order to, so what we wanted to do was to collect also a large-scale population genomics data set connected with the phenotypic data. So we set off in 2017 to make a large collection of foraging bumblebees from Colorado, together with our collaborators, and we collected around 650 samples, and also used them to measure body size and tongue length. And they were collected from this set of sites in Colorado. So a variety of different regions on different mountains. And here you can see a member of the team, Julia Jones, up in the mountains doing some collecting. So the first thing we did is to produce the genome assemblies of these two species. They have 250 megabase genomes, which is quite convenient for genome assembly, fairly small sized. We used Oxford nanopore long read sequencing as the sort of backbone for these assemblies. This was coupled with 10x chromium linked read sequencing, and then RNA sequencing to in order to for producing annotations. So the 10x chromium linked reads based on Illumina can be used both for error correction of base quality, but also for linking together contigs, and we used a variety of different bioinformatics packages to deal with this. So we think we produced a pretty high quality genomes from this using this method. We placed them onto pseudochromosomes using the genetic map from Bombus terrestris, because we didn't have a genetic map from these assemblies. And so this produced assemblies with pretty high contiguity. And just as a comparison, if you compare this with the assemblies of some previously generated species using shorter read technologies, you can see that the lengths of the contigs here, the M50 is about 100 times higher in these long read assemblies compared with the more sort of older fashioned methods. So we think these are a pretty good resource going forward. The next thing we did was to do Illumina sequencing of the samples that we collected that were, and these were done using Illumina HiSeqX. So that just a quick overview of what that data looked like, as you can see here. And what you can see from this is that so these are the trees that are made from the two species that we collected, although we quickly realized that it seems more likely that there are actually 3 species. So you can see that the Bombus balteatus samples are found in one cluster, whereas the Silvicola samples actually found in cluster into 2 different clusters, and that there's very little substructure that can be observed according to geography. These 2 clusters are found in all of the locations where we collected these bees. So it seems like within each species there's very little geographical clustering, but this additional species, which seems to be indistinguishable morphologically from Bombus silvicola, and we call it Bombus incognitus. That one is found in the same locations where we also found silvicola. And here you can see a principal components plot, where you can see that these silvicola samples are very distinct from the Incognitus samples. In the morphological analysis that we've done, we haven't been able to identify any distinguishing characters, or there are still things that are possible to look at. And we see no intermediates between these different samples. We also there are no other sort of known species that are good candidates for being the same as this. And so there isn't genome-wide data for all other species within this subgenus, but there are sequences from individual genes, and this is a tree that's produced from the PEP-CK gene, which is commonly used in these kind of studies, and you can see that here is our Incognitus, which clusters separately to a lot of other species within the group that are kind of potential candidates for being the same one. So it does seem that this is distinct from samples that have been analyzed before, and we see a similar pattern using the CO1 gene. So we find that although we didn't find any kind of definitive differences between these two species, we do find that they were found at slightly different locations. So there may be some ecological differences, but there's nothing that's completely categorical difference between them that we've seen so far. So this will be interesting to look at further. So after this discovery, we decided that one kind of interesting thing that we wanted to look at was what's the history of speciation between Bombus sylvicula and Incognitus. So what can we learn by comparing these genomes about the history of speciation and whether gene flow occurred during this process. So I'll talk a bit about that study next, and then I'll get back to what we've found regarding the phenotypic evolution. So just to sort of give you some background about the speciation genomics in general. So a kind of a common model that's been used to understand speciation with gene flow is that it's predicted that certain parts of the genome become isolated early on, and then by different possible mechanisms they expand over time as two species diverge. And so gene flow can occur in the beginning between most of the genome, and then at the end you have two completely isolated genomes between two species. And these regions are predicted to be regions that are involved either in ecological specialization or that they're involved in intrinsic genomic incompatibilities like Dubchansky-Muller incompatibilities. And it's been a pretty popular thing to do to scan the genomes of fairly closely related species in order to try to understand what the genetic basis of these barriers to gene flow are. And but it actually it's been pointed out that there are actually multiple explanations for why you can have these peaks of divergence. So there've been, and one of those explanations is that they're islands of speciation that are resistant to gene flow. Another possibility is that they're regions of low recombination that diverge between populations due to the interaction between selection and the recombination landscape. And there have been a number of studies of different species that have identified parts of the genome which using this method, which are do seem to code for incompatibilities between species or due to genes involved in ecological specialization, for example, differences in beaks. But also a number of other studies have also identified this landscape where it seems to be mainly due to the interaction between selection and recombination rather than having something to do with speciation and gene flow. So we wanted to use these theories to analyze what we can learn about speciation in this new cryptic bumblebee species. But one way to distinguish between these two models is to use the statistic dxy, which is absolute divergence. So if you look at the left-hand graph, what you can see is that the model of when you have some regions which are prevented from undergoing gene flow because of barriers to gene flow. And in this example, you have regions that are undergoing linked selection. The way to distinguish between them is to use this absolute divergence, or dxy statistic, which I'll be coming back to later. So this is a useful way of uncovering the mechanism of species divergence. So what we have in our situation is we have a couple of fairly closely related species that are found in sympatry, and we're able to compare the genome variation in these two species. And those we collected in Colorado. But then we also wanted to have a control, and we were able to get some published data of another pretty closely related pair of species, which are found in allopatry. So their ranges are mainly not overlapping and have had less opportunity for gene flow in their history. So these were the two comparisons that we wanted to do in order to understand the differences in genome divergence in a pair of species where gene flow is possible and another where it's probably less likely. This is a tree of all of the sequences that we've put together. On the left, you can see the samples we collected, and on the right, you have the control samples from published data. So then we can start doing genome comparisons. So this is the sort of the first slide with a lot of data, which I will talk to you about. So what you can see here is the distribution of FST across three different comparisons. So the first is when we compare genome variation in populations of the same species from different locations. The middle panel is the two species that we found in sympatry, and then finally the bottom one of the two species that are found in different locations. And I think what you can see is that there are definitely quite distinct peaks of divergence across the genome. So there are peaks which characterize differences between them, and that these peaks tend to occur in pretty much the same location in all of these different comparisons. So it seems to be that there's something inherent in the architecture of the genome that's causing the sort of same regions to diverge faster in multiple sort of independent comparisons. And as has been found before in sort of similar studies, we can see that when we make a map of recombination rate variation and identify centromeric repeats, that those features do tend to correlate fairly well with the peaks of divergence that we observed in these genome comparisons. So kind of at first sight, this seems to match the model that I mentioned earlier about how link selection can cause genome divergence. And this is just another graph to show that as you move away from these the center of these islands of divergence, you move from lower recombination, and that increases. And you can see similar patterns when you look at GC content and the amount of repeat sequence. So it seems that they're centered on regions of low recombination and centromeric repeats. But even though these islands occur in similar positions in the different comparisons, we can see that there are some differences in the distribution of divergence. One of those things is that if you look at the distribution of FST in windows across the genome, you can see that in this sympatric comparison, it's a bimodal distribution in contrast to the other comparisons. And what this suggests to me is that there's a sort of a subset of regions with particularly high divergence, suggesting there's maybe some kind of additional force, such as, for example, differential gene flow, which causes one part of the genome to be particularly divergent. Another thing that we can see is when we look at this DXY statistic, or absolute divergence, this is shows a very different pattern if we compare the sympatric and the allopatric comparison. So in the sympatric comparison, we can see that DXY is significantly elevated in our islands of divergence, whereas in those same regions, it's reduced in the allopatric comparison. So this shows that there's possibly two different processes going on. And as I kind of mentioned earlier, this elevated DXY is a fairly robust signal of differential gene flow, which suggests that maybe these regions are acting as barriers to gene flow in the sympatric comparison, or in the history of their divergence. In addition to this, we can also see that there's a fairly extensive reduction in nucleotide diversity around these islands of divergence, which is much stronger in the sympatric than the allopatric comparison. So it seems suggests that there's kind of a more intense level of link selection affecting these regions compared with the allopatric comparison. So it seems like you have a more extensive and intense reduction in nucleotide diversity in the sympatric comparison in these islands of divergence. Just to sort of put these different findings together and what my interpretation of them is, we can see that in both of the in the sympatric and the allopatric comparison, we found these peaks of divergence, which tend to occur around centromeres and in regions of reduced recombination. And this is fairly typical of what's expected when you have link selection driving, interacting with the landscape of divergence, driving landscape of recombination, driving divergence. But we saw that there's some differences between the characteristics of these islands of divergence in these two comparisons. in particular, the DXY seems to be elevated in islands of divergence in the sympatric comparison, and FST is bimodal. And so this suggests to me that you have a signal of differential gene flow in the sympatric comparison. So when there's there's been opportunities for gene flow in their evolutionary history, and that these genomic islands have acted as barriers to gene flow, or that it's occurred at a much lower extent in those regions, whereas the allopatric comparison have been more isolated by geography. So, I mean, what this suggests to me is a sort of a way to kind of put these two different ideas together to incorporate them. So we have features of these islands of divergence that match both of the possibilities, that they seem to be regions that in the sympatric comparison are resistant to gene flow, but also that they are accumulating in regions of low recombination. So it suggests that we have sort of features of both of these, and that kind of suggests that maybe you have kind of potential incompatibilities that occur across the whole genome, but they're becoming fixed more readily in regions of low recombination. And there are kind of some other interesting studies that have come out fairly recently that kind of match this idea in the sense that it seems that the levels of gene flow that have been inferred between populations of hybridizing species seems to correlate with recombination rate. So it seems that so one explanation for that could be that regions of low recombination are important in generating incompatibilities between species. So now I'll just summarize quickly this first part, in that we found that barriers to gene flow between these two related species seem to have accumulated in regions of low recombination near centromeres. Suggests that potential genomic incompatibilities could be quite widespread in the genome, but they become fixed more readily in these regions. So suggesting that the architecture of the genome is pretty important in determining which parts of the genome act as barriers to gene flow. And one kind of speculation that we have is that of kind of the sort of why this of a possible scenario of how this speciation occurred is that maybe you get periods of warming where the habitats become more fragmented, leading to sort of temporary isolation at higher altitudes, and then there's periods of secondary contact where gene flow can occur, but divergence proceeds. And I just made a simple cartoon to just sort of illustrate that idea, that kind of in warmer periods you can have temporary isolation, and then followed by when you have sort of partial barriers to gene flow have developed, you can have secondary contact, but that divergence still continues. So this could potentially be a important mechanism in bumblebees. And just to sort of take this a little bit further and speculate a bit more now that we're doing a lot more sort of population genomics is, I mean, I was quite surprised to see that you have these genetically distinct clusters. And I've also noticed that, and that they weren't possible to detect morphologically before. So it's kind of interesting to me that whether we're likely to find more cryptic species from doing these kind of large-scale population genomics surveys, and there are certainly examples of how these kinds of findings are coming out in recent times. So I think it's interesting to speculate about how many more cryptic species might be out there when we start looking at biodiversity more in this way. I've also had some interesting discussions about whether evidence from DNA is even enough to define a new species, if you need to have some functional expression of that as well. And another thing I would like to sort of note from this is these species are pretty well separated. We estimate it's up to sort of 400,000 years ago, but that they weren't detectable from barcoding. So I think it kind of exaggerate, or it sort of argues towards the importance of genome sequencing where it's possible. So that was what I wanted to say about the first study. And the time that's left, I'll just briefly go over what we've learned about the genetic basis of phenotypic evolution of these species. So as I mentioned before, the sort of the impetus of that got me interested in this population was this decreasing tongue length, which seems to be a important kind of process that's going on that can be important for biodiversity. And so one of the what I was interested in looking at was whether these changes are due to natural selection or plasticity, and whether they're what the genetic basis might be. And one of the kind of inspiration for wanting to look at this came from kind of studies that I've been partly involved in on Darwinsfinches, where a species of Darwinsfinch in the Galapagos been studied over many years, and showed that the size of the beak had been slowly changing over years. And in this case, it was possible to identify a gene with a very large effect, where genotypes at this gene correlate quite strongly with differences in beak size. And also it was possible to show that survival during a period when there was a large drought, and the beak size dropped a lot because of it was necessary to switch to different food sources. The survival in that period correlated strongly with the genotype. So I was interested in whether kind of similar things could be seen in the example of the bumblebees. So just going back to the data set that we had about 650 genome sequences from these species that were initially believed to be 2, but we think are 3. And we also had phenotypic measurements from both the intertegular distance, which is a proxy for size, and the tongue length of these samples. So one kind of initial observation we made was that it seems that among the samples that we collected, they were already shorter than the ones that had been collected only a few years previously. So just before I get into that, what you can see here is that before we thought there was only Bombus sylvicula. Now we think there's an additional species, but those are not different in the tongue length. So it seems that differences in their abundance are unlikely to affect these analyses. But anyway, what we can see is that that there seems to be an ongoing decrease in the lengths in both of these different species, and that seems to occur on in all of the different mountains that we sampled where there is data available. So it seems like this trend is ongoing. However, unfortunately, we weren't able to find any strong correlations between particular genetic loci and phenotype using a genome-wide association study of using these in these samples. These show just QQ plots of the correlations between the different phenotypes and the genome sequences in these two different species, and you can see that the observed and expected p-values across the genomes are a fairly straight line. And so this is for the glossal length, the tongue length corrected for size, the intertegular distance, and the intertegular distance on its own, with one exception, and that's this one where the you seem to have some more significant p-values, and that is a correlation that we observed with intertegular distance and a SNP in Bombus sylvicula. So that's that SNP here, and it's hard to say whether this is a true hit or not, but it definitely seems to have a high significance, and it's found in a gene which kind of makes sense that it might be involved in size, because it's involved in the production of an enzyme which is important for digestion of pollen. So it has a role in nutrition. So it seems that there's variation in this gene which may affect body size. But in kind of contrast to the results that we've from, for example, the Darwinsfinches, where there was a very strong effect from a single gene, it doesn't seem to be the case in this bumblebee system. Another thing that we could do is to try to estimate the heritability of these traits, and that can be done using an approach called Gremel. But unfortunately, this approach needs quite large sample sizes in order to detect heritability. So we couldn't really confirm that it had a strong genetic component using this technique. Basically, the idea is that to correlate between variation in kinship in the sample and covariation in phenotypic measurements. So when we include samples which we infer are likely to be from the same colony, we can see that they do seem to have significant correlation in size, but in that case, it's harder to rule out shared environmental effects. So when we exclude those samples, it's harder to detect significant heritability. So the heritability of this tongue length and the tongue length trait seems to still be unclear. So just to summarize the second part, we can see that we have pretty high connectivity in between the mountains, so there's very little geographical substructure in bumblebee populations. And we observe that these sort of tongue length changes that were observed previously are ongoing. Our kind of analysis of the genetic underpinnings of these changes are sort of inconclusive. We can see that we identified a SNP that seems to affect body size, but it seems that the genetic basis of this tongue length trait is likely to have fairly complex genetic and environmental components. But we're hoping to investigate this more and look at by looking at some of the more ancient the historical genetic samples which have greater variation in phenotype, and to look at the evolutionary changes that have occurred over time. Yeah, just to conclude, we infer that there's a cryptic species amongst our samples which hadn't been observed before, and which we can't find any, or haven't so far found any morphological differences between Silvicola, although we infer that it diverged around 400,000 years ago. We also infer that it likely evolved under conditions of gene flow between Silvicola and Incognitus, and that there are polygenic, or that there are barriers to gene flow have accumulated mainly in regions of low recombination and centromeres. And then finally, we can see that within these populations, we have this ongoing reduction in tongue lengths, but we're still sort of unable to determine whether they're have a genetic or a plastic basis, whether they're due to natural selection. But that work is ongoing. So just to kind of wrap up, I thought these species have a sort of a circumpolar distribution, and there's it seems there are a lot of interesting questions regarding the diversity of these species. So for example, you have species that are found in completely different continents, and it's difficult to determine whether there are whether they're different species or not. whereas you have, it seems there are also cryptic species found in the same locations, which are difficult to distinguish. So there's a lot of kind of interesting evolutionary processes and going on there. So I think there's a lot of kind of open questions about this these set of species in terms of how many there are, whether they can interbreed, what the sort of mechanisms of speciation are, and also whether these sort of morphological changes that have been observed are something that happens in more than one population, if they're a general feature of what's going on under climate change, or if it's a sort of isolated example. So all those things are kind of things that are interesting to look at in the future. So just before I finish, I would like to acknowledge the people that worked on this study, in particular Matt Christmas, who spent some years doing the bioinformatics work, but also the rest of the team in Uppsala that worked on the collections and lab work and bioinformatics. Alexander Su and his group that worked on the telomere analysis, and then Nicole and Jennifer in the US, who are our collaborators that kind of did the initial study on the tongue length decreases. So thank you very much, and I'm happy to take questions.